Hey everyone, welcome back to the Biochem Words by Hammad and welcome to the lecture of our drug discovery and designing series. Today's topic is one of my favorite where biology shakes hand with computer. And Imagine discovering a new medicine without mixing chemicals, without even touching a test tube, just you, your laptop, and a little bit of digital magic. That's what in silico drug discovery is all about. Think of it like Netflix recommendations. The system predicts what you will like. In our case, software predicts which molecule might click perfectly with a disease target. Pretty cool, right? So move forward. Our what is drug discovery? It's basically detective work. We are trying to find which tiny molecule can fix a biological problem, like finding the right key for a stubborn lock. In daily life, think of your mobile charger. Not every charger fits every phone, as there are a lot of kind of chargers like iPhone charger, Type C charger, USB 2.0 charger. So not every phone fits with every charger. Only the right one works. Similarly, not every molecule works with every protein. The fit has to be perfect. Traditionally, scientists tested thousands of molecules in the lab. It took years and cost billions. But now we can simulate this process virtually on computers, screening millions of molecules in hours instead of years. So, the core paradigm is the target, then the lead, then the optimization, then the trials. So, drug discovery has all these steps that we'll discuss in our lecture with a short introduction, and in our next upcoming lectures we will explain them. In every lecture we cut a single step for every lecture, like target identification, drug discovery, lead discovery, and lead discovery, preclinical trials, preclinical testing, clinical trials, and now it's all about when you are following us and entered in the series of computational, then you are transitioning from traditional, the experimental work that you can do in the lab, go to the lab and work in the lab, in the class in the lab. That's called the in vitro working. And so you are transitioning from traditional methods to the computational. One of the thing that you have to consider is there are three terms. Here's the short, I add it up, that one is in silico, then other is in vitro, and the third one is in vivo. These three are different kind of researches, different ways of researching. They are named on the way of researching. Like in silico means, in a short term, I usually when I'm delivering my lecture, or discuss with anyone, I only give them a short term of these things, like what is drug discovery, and it have three kinds of research, like in silico, in vitro, and in vivo. In silico means in computer, in vitro means in lab, and in vivo means in life. Like in silico, you are doing your research on computer, you are doing your workings on computer. Like in computer, the research which is done in the computer is in silico. In vitro means in glass or in lab, you are doing your research in lab or in the petri dish, in a glass, is the in vitro. And the number third one is in vivo. You are going through the clinical trials, preclinical trials, and clinical trials that we'll discuss in our next slides. So move forward. The journey of a drug, while you are doing the drug discovery research. So these are the steps that include in the journey of a drug. Let's walk through the journey of every medicine sitting in your cabinet. Whether it's a painkiller or an antibiotic, each one went through this exact process. Number first is the find the target identification, and the protein, enzyme or receptor causing trouble. It's all about you have to uh, find out that which protein is causing the trouble, which enzyme is defective and causing the trouble, or any external pathogen that have entered in our body and affecting which protein and which enzyme. So this is the target identification. First of all, we have to identify in our body, in the body of the individual, that which thing is causing problem. So first of all, find the target. Number second step is validate that it's really the bad guy. Validation is also mandatory, that we have to validate that it's causing problem or not. Rigorously confirm that modeling the chosen target truly produces the desired therapeutic effect in cellular or animal models, and that the target is druggable. That we are using the target, the protein, the enzyme is also druggable or not. So, the third one is find a hit, the first potential molecule that shows activity. We have to screen a large number of libraries. In our next lectures, I'll show you that which kind of the libraries you have to make, and how you can make the libraries of compounds and then screen them by doing the docking tools, utilizing docking tools like Autodock, Pyrex, molecular operating environment. So these are to utilize the hit identifications. And number third one is optimize it, tweak the molecule to make it stronger, safer and smarter. We have to investigate about their ADMET properties. ADMET properties means absorption, distribution, metabolism, excretion and toxicity. These all properties are related to our body, that even though this compound we are using, when we accumulate it or when we take it, how it absorbs in our body, how it distributes in our body, and how it can be metabolized in our body, and how it would be excreted out of our body. And also the main thing is the toxicity. Is it toxic or not? It will cause any toxicity in our body or not? So this is often the most iterative phase driven by medicinal chemistry design cycles. So after observing all these properties, we have to optimize the compound accordingly. And finally test it in living systems, preclinical and clinical trial. You first design, test, prototype, fix the bugs and only then launch it. So think developing a new smartphone. So same logic, but instead of gadgets we are engineering cures. The comparison between traditional versus in silico drug discovery. So here's the classic face off. Traditional drug discovery is like cooking without a recipe. You keep experimenting until something tastes right. And in silico drug discovery is like using artificial intelligence in the kitchen. It predicts what ingredients will make the perfect flavor before you even start. So with powerful algorithms, we can virtually test thousands of molecules, saving time, money and effort. As you have a number of compounds, you have to spend a lot of time in the laboratory doing the wet lab. You have to go through all the compounds one by one, and also this one is cost effective, and also it demands a lot of time, that mostly it takes 10 to 15 years from start to market. So but in silico is like 15 years and billion of dollars versus a few months and a laptop. Which one would you choose? So these are the comparison that I have made. Time frame, as I have explained, that it's long time frame. It needs a long time period to do the research, but significantly shorter. Rapid compound iteration and risk mitigation. Cost, it's high cost, that even the compound we have to purchase it, but it's not effective, but we have purchased it, we have to work through it. But when we do the in silico, you don't have to purchase the compound, but you have to work on the computer and work through it and go through the working, and then you will screen the compounds, then which will be the effective, you only buy that one and then validate in your wet working. So that's the cost effective. They will also save the cost in our traditional lab. Screening method: experimental high throughput screening of physical chemical libraries, virtual screening through docking, pharmacophore modeling. Docking is the main procedure that moves around the computational chemistry. Even you are going through the drug discovery or any other research through the computational, docking is the main procedure that everyone should have to learn. So in our next lecture we'll make sure that you will learn it and be expert of docking. So initial accuracy focus, it's broad rational focus screening based on molecular properties and target structure. So move forward, bioinformatics and chemoinformatics and the dynamic duo. Uh, every superhero has a sidekick. Batman has Robin, Sherlock has Watson. In drug discovery, bioinformatics and chemoinformatics are that dynamic duo. Bioinformatics deals with protein and DNA. It tells us where the disease lives. It basically works on our target, that the target we are selecting, by hitting selecting the target, it helps us is the one that is bioinformatics, that utilizes the web servers like RCSB PDB, where we can get the protein of every virus and published, those are published. And the blast for the protein sequence, uniprot for also protein sequences, and blast for the we have get the protein, and then you can do the blast on the NCBI and get the same protein structures like it. And the chemoinformatic handles molecule, it tells us which compound might fix it. It also goes through the web servers like PubChem, Campbell, and drug bank, where you can get every compound. You just have to write it, the silymarin and ethanol, any compound you can write it, they will give you the 3D structure of that compound. So when they work together, bioinformatics and chemoinformatics, we can rationally design a drug rather than guessing blindly. It's like using Google Maps to find the fastest route instead of wandering around the city hoping to reach your destination. So moving forward, essential components of computational research, major tools in drug discovery. Now it's like the Avengers of computational world. Avengers. I'm a big fan of Avengers. I'll give you the examples like this: Harry Potter, Avenger, Iron Man. And now meet the Avengers of computational world. NCBI and Uniprot help us find the protein target. Number first is the target identification. NCBI and Uniprot will help us to find protein target, the bad guy in our story. So number second is compound libraries. How you can make the compound library, as I have discussed before, that PubChem, drug bank are our molecule libraries. Imagine them as molecular supermarket. You just go to the supermarket and get your uh, as you are wishing that way of the compound, you can get them there in the molecular supermarket of naming PubChem and drug bank. And number third is affinity prediction. Autodog and Pyrex do the docking. Molecular operating environment also can do it. The virtual handshake between the protein and the ligand. And drug likeness and admit, Swiss ADME, PKCSM, and also the Admit Lab 2.0. It's also a web server that will use to be to predict whether your molecule behaves like a drug, soluble, safe, and stable. And Chimera or Pymol that are utilized for your screening and visualization. I show you I have opened them in the sidebar. You can, that's the Pymol desktop. You open Pymol like this. I show you a protein. I have saved the structures. There's a project. Yes, I have the protein. That's one. In which format it is? It's PDB. So open. That's how it can be visualized. Now move it. You can move it. It's 3D structure. You are seeing it, and you can also zoom in, zoom out, and here you can show wire lines and like label, hide, anything, color by chain or element. You can say by chains, it has 2 chains. So we'll discuss that in our upcoming lectures elaborately. And the other one is also for visualization is Discovery Studio. As you are seeing, here's the Discovery Studio. You can also open it like this. Here you can see it's a vast server where you can get every knowledge about the, and also handy to everyone, protein groups. And there is a data table also, the molecule, the cell, amino acid chains. It have 2 amino acid. Every amino acid will show you that how much amino acids are there in the protein, and in which color they are labeled, and atoms, the which atom they are containing, and the bond they are containing, like nitrogen, carbon, carbon, oxygen, and group. So that's the Discovery Studio. And here also the Autodoc tools from the MGL tools. Here you can get the preparation of your ligands and protein receptor molecules for that could be readable, and also it is used to optimize the compounds. As you have made the library, you have done the docking, but it's giving the compound like it's toxic or not soluble, then you add up the bonds or optimize it here. And also you can done optimization in the do optimization in the Discovery Studio. Here it is. And Pyrex, it's also used for the docking for number of ligands you have. You have 100 of ligands library. You just add the library here one by one, or whole library you have, you put it here, and all the compounds will be opened here, and you can do the docking. Same, it also uses the Autodoc Vina for its working. So these are the softwares, as I have given you the general overview of this software, and we'll discuss them in our upcoming lectures. So move forward. Live demo of doing the molecular docking. That's the ligand retrieval from PubChem target retrieval from RCSB in the PDB form. I mentioned the VP35 protein of Ebola virus. That's my previous working I have done on it, so that's why I have mentioned the VP35 protein of the Ebola virus. So, and also the structure visualization, I'll also add up a video, a short video here that will visualize you that how the docking process goes through all this, and so you will see the video.
As you can see that how the molecular docking process occurs. Here you have to make a separate folder on desktop and save your protein, and by deleting all the extra elements or compounds from your protein, and also the your compound in the same figures, like it's in the file format PDB, then open it up in the MGL tools Autodoc, and then also change the file type and optimize it. Then you have to run through the Autodoc Vina. As you have seen, it's a short video. I have added a screen recording of the process that is done here. And sometimes you got a glitch that you have saved a compound in the PDB format, but it's not opening in the next software, because most of our things, like we have to always think about that which software accepts which file type. So that's a very precious thing, that Discovery Studio can open up every kind of file type, but Autodoc MGL tools will only read the PDB and PDB Qt files, and the Vina we are running is only made to read the PDB Qt files. And also there are some requirements along with the compounds. We have to make the configuration files that will give the dimensions to the Vina, that on which dimensions of protein we want it to work. As you have seen, it's running. We have just to paste the codes. And
so coming back to the lecture, the In-Silico workflow, as we have previously discussed, the ultimate recipe of digital drug discovery. Step 1, identify the target. Step 2, find the ligand. Step 3, prepare structures. Step 4, run the docking, virtual binding. And step 5, admit prediction. Step 6, analyze result. Think of it as online matchmaking. You find a perfect pair based on compatibility, appearance and behavior. Except instead of dating, we are talking about molecules and proteins. So move forward. Real world applications, impact and future of our drug discovery in silico methods. These methods aren't science fiction, they are happening right now. Most of the researches, as you will go through, will include the in silico. Also, they done the in vitro, but the in silico research papers are now coming out in bundles. So COVID-19 messenger RNA vaccines were modeled using computational prediction of spike protein behavior. So docking studies are used for cancer drug design, like inhibitor for tyrosine kinases. These are the examples of our recent researches in our world that are utilized by in silico methods. As the CRISPR gene editing, you will see the cancer treatment as scientists done. You go through the Facebook and you will see such a post if you have FYP of seeing these things on your mobile phone. Even in neurodegenerative diseases like Alzheimer, scientists simulate which molecules cross the blood-brain barrier. So yes, the same computational logic you use when your phone predicts your next word is now helping save life. And limitations, it also have limitations. Now, before we get too carried away, a quick reality check. In silico results are predictions, not proofs. The computer might tell you a molecule fits perfectly, but in the lab, it might not even dissolve in the water. So, computational work narrows down possibilities, but the wet lab experiments still seal the deal. It's like the Google Maps suggesting a route, but you still need to drive it yourself to know it's there if there's a traffic jam. And summary, let's wrap up. Today we decoded what drug discovery really means, explored how computational tools transform the game, and even walked through databases like PubChem and RCSB PDB. You now understand how molecules virtually date proteins before heading to the lab. Remember, your computer is now your lab assistant. Treat it well, and it will help you discover molecules that could save millions of lives. And in our next adventure, we'll go deeper, learning how to identify and validate targets using bioinformatic tools like NCBI, UniProt, and String. So make sure your systems are ready, your coffee is strong, and your curiosity sharper than ever. Because for next lecture onward, we truly begin designing drugs in silico. So thank you so much. If there is any question, you can ask me in the comments, and don't forget to like and share with your fellows and your well-knowns, and follow us on the Facebook with the name Hammad Bashir Khan, and subscribe us on YouTube. It's Biochem Words by Hammad, and thank you so much for watching. We'll see you in the next lecture. Thank you so much. Allah Hafiz.